Hello everyone. Today, let us see some question answers from this chapter. Methods of biotechnology that man adopted and practiced traditionally. Answer is, yeast was used to prepare food items like bread. Bacteria and fungi were utilized to convert sugar into alcohol or acids. Practice the method of selecting and rearing of cattle or crops of superior hybrid variety. Give example for modern biotechnological practices. Development of human insulin producing bacteria. Production of farm animals that yield in medicines or vaccines. What is modern biotechnology? Modern biotechnology includes the production of organisms with desirable qualities by changing their genetic material. What is genetic engineering? Genetic engineering is a technology that controls traits of organisms by bringing about desirable changes in their genetic constitution. How is it possible to bring about desirable changes in an organism? Genetic modification in organism is done by cutting or joining specific genes using certain enzymes. This process is known as genetic engineering. Describe the stages in the production of human insulin bacteria through the process of genetic engineering. Answer is, from human DNA, cut the gene responsible for the production of insulin. This gene is joined with bacterial DNA, which is used as the vector. Insert the joined DNA in the bacterial cell. Define vectors in genetic engineering. Vectors are other DNA, usually bacterial DNA, by which genes can be transferred from one cell to another. What do you mean by genetic scissors and genetic glue that are used in the process of genetic engineering? Answer is, the enzymes like restriction endonuclease used to cut DNA at specific sites are generally called as genetic scissors. The enzymes like Ligase used for joining DNA at specific sites are generally called as genetic glue. Next question is genetic scissor. Restriction endonuclease, genetic glue at dash. Answer is ligase. What is DNA profiling? The technology of testing the arrangement of nucleotides in the DNA of person is called DNA profiling or DNA fingerprinting. Next question is the developer of DNA fingerprinting. Answer is Alex Jeffrey. Next question is, what is the basic principle behind this technology? The arrangement of nucleotides in the DNA of each person differs. In DNA profiling, we test the arrangement of nucleotides in the particular person with that of others. Next question is, mention the scope of DNA testing. Answer is, DNA fingerprinting is helpful to find out hereditary characteristics, to identify real parents in the case of parental dispute, and also to identify persons found after a long periods of missing. Next question is, genetic engineering caused tremendous changes in medicinal field. How? Answer with examples. Answer is, human insulin producing bacteria, farm animals which produce human insulin and growth hormones, medicine producing plants. What do you mean by farm animals? Answer is, genes responsible for the production of human insulin and growth hormones, etc., are identified and inserted in animals like cow or pig to transform them into farm animals. Animals providing pharmaceuticals or medicines. Medicines thus produced can be extracted from the blood or milk of such animals. Next question is, the sum of genetic material present in an organism is called dash. Answer is genome. The human genome includes about 30,000 genes present in its 46 chromosomes. Define the term junk genes. Majority of our genes seen in the chromosomes are non-functional and is known as junk genes. What is Human Genome Project? The human genome includes about 30,000 genes present in its 46 chromosomes. The secret of human genome is revealed through a project known as the Human Genome Project, started in 1990 and ended in 2003 in various laboratories of the world. The gene mapping technology helped us to identify the location of a gene in the DNA. What is gene mapping? Gene mapping is a technology by which we can locate a specific gene in the DNA responsible for a particular trait. Next question is, what is gene therapy? How is it beneficial to mankind? Gene therapy is the method of curing genetic diseases by removing disease-causing genes from the genome and inserting normal functional genes. Gene therapy is beneficial for the sustenance of humankind. DNA profiling for identifying person or other organisms. Dash for the treatment of genetic diseases. Answer is gene mapping. How is genetic engineering favorable to protect the environment? Genetic engineering is beneficial for neutralizing substances that cause pollution to nature. Petroleum digesting superbugs bacteria are example. Next question is, mention how gene technology becomes beneficial for our sustenance. Answer is organisms producing insulin and other medicines. Remedy of genetic diseases and gene therapy. Productive and disease-resistant varieties. Food crops. DNA fingerprinting. Conservation of nature. Explain briefly about the merits of genetic engineering in the field of food and agriculture. Answer is, genetic engineering influence in the production of genetically modified, disease-resistant and high-yield varieties of animals, food crops and cash crops. Next question is, what are the possibilities to misuse gene technology? It is criticized that genetically modified varieties are too indigenous varieties and may cause health issues to humans. There are possibilities to use the genetically modified organisms as bioweapons. What are bioweapons? Which is the technology behind biowar? Bioweapons are genetically modified pathogens that might be applied any country to their enemies. Genetic engineering is the technology behind this kind of biowar. Next question is, make a few logo sentences that can be used for the awareness program against the misuse of science and technology. Answer is, genetic modification can be allowed only for the benefit of mankind. Avoid all weapons including bioweapons, save life. Science and technologies are meant for protection, not for destruction. Thank you.